Hey, good afternoon, everyone. My name is Dr. Eric Toring. I'm the Senior Vice President for the American College of Veterinary Preventive Medicine, and I want to welcome you to today's webinar. The webinar today is sponsored by the American College of Veterinary Preventive Medicine and our partners, the American Association of Food Safety and Public Health Veterinarians, the National Association of Federal Veterinarians, the U.S. Animal Health Association, and the National Association of State Public Health Veterinarians. So it's my pleasure today to introduce our two speakers. First of all, Dr. Rebecca Fallender is the Senior Wastewater Epidemiologist in Oregon. She received her veterinary degree from Tufts University in 2016 and practiced as an emergency and critical care veterinarian in Portland, Oregon for several years before obtaining her master's in public health from the University of California, Berkeley in 2021. Dr. Fallender completed a 2-year public health fellowship at the Oregon Health Authority before accepting her current role. Her interests include wastewater-based epidemiology, emerging infectious diseases, study design, and bioethics. Also joining us today is Dr. Ryan Scholes. Dr. Scholes is a graduate of Oregon State University, holding a DVM and a master's in public health from that institution. In 2020, Dr. Scholes was appointed as the state veterinarian for the state of Oregon. In this position, he serves as the Chief Livestock Disease Control Official, overseeing animal health, regulatory compliance, and emergency management. So welcome, Dr. Fallender and Dr. Schultz.
Thank you so much for that introduction. Can you hear me okay?
I can hear you just fine.
Great. And I apologize, my voice has been going in and out a little bit this week, so hopefully it'll hold for this presentation. But I'm going to be going through most of the slides in today's presentation, and at a couple of points I might ask Dr. Schultz if he has any details to add, and hopefully he'll jump in. And then, yeah, like Eric said, we'll both be available at the end to answer questions. So today's presentation is on avian influenza A H5 surveillance in Oregon. As an outline, I'm going to give a brief review of the emergence of the new genotype of avian influenza H5 and a broad overview of what avian influenza surveillance looks like in Oregon. This will include the addition of our wastewater surveillance program for the avian influenza H5 subtype. And then lastly, I'll go through the first case of avian influenza 2.3.4.4b that was detected in pigs here in Oregon. So just to review some avian influenza terminology, I'm sure most of you here are familiar with this. Feel free to tune out. But influenza A viruses commonly circulate in humans, waterfowl, domestic poultry, swine, horses, dogs, and bats, but infect many other species with more limited transmissibility. So the term avian influenza refers to the viral disease of birds caused by influenza type A viruses. The disease varies in severity depending on the strain and species of animals affected. Our highly pathogenic avian influenza, usually H5 and H7 strains, have high mortality rates in domestic poultry, can wipe out entire flocks in less than a week. Low pathogenicity avian influenza strains occur naturally in wild birds around the world. They typically cause a mild or asymptomatic illness in domestic poultry. However, some low-path avian influenza strains can become high-path in poultry. Avian influenza rarely infects people. Historically, transmission to people has been through direct contact with infected animals or contaminated environments, and human-to-human transmission has not been documented. So starting in 2020, a new genotype of highly pathogenic H5N1 emerged in Europe. This new genotype infected large numbers of wild and domestic birds across Europe and quickly traveled via waterfowl flyways to other countries. This new panzootic genotype was characterized as clade 2.3.4.4b. In January of 2022, the USDA APHIS announced the detection of the new genotype in the United States. It was an American widgeon in South Carolina, and it followed a detection in Canada a month earlier. Detections of clade 2.3.4b started sporadically in the Atlantic flyway, shown in black on the map on the right, and then quickly spread flyway to flyway to the Mississippi, Central, and Pacific flyways. Rapid expansion of cases occurred in the spring of 2022, and the first detection in Oregon was in May. This map, updated in November 2022, shows detections in wild birds, domestic poultry, and mammals across the United States. There have now been at least 6 separate documented introductions of clade 2.3.4.4b into the migratory wild birds in the North American flyways, genotypes A1 through A6. And during its spread, the panzootic genotype has undergone further reassortment with local low pathogenicity avian strains, resulting in diverse lineages. Today, the USDA reports over 18,000 detections in wild birds since December of 2021, with almost 700 detections in Oregon. For domestic birds, Oregon has had 3 affected commercial flocks and 58 affected backyard flocks. So the number of high-path avian influenza detections in wild birds in Oregon is not surprising. Oregon has an incredibly high density of migratory waterfowl. Oregon is located along the Pacific Flyway, which stretches from Alaska to Patagonia. In the summer, birds intermix in the Alaska-Yukon area. And in the fall, the Pacific Flyway is one of the most populous, containing 100,000 birds a day flying south. Also, Oregon contains several significant stopover areas for these birds during their migrations. So this genotype, as you guys know, has also shown a high propensity to infect mammals. And in March 2024, 2 years after it spread in birds across the United States, the U.S. Department of Ag reported a multi-state outbreak in dairy cows. So phylogenetic analysis suggested that clade 2.3.4.4b entered the dairy cattle population via an introduction from an avian host several months before it was detected in a cow. As of this week, the number of confirmed dairy herds affected is over 1,000 in 19 states. Infections in cows have disproportionately affected lactating dairy cows with high viral loads in the milk of infected cows. There has not been a confirmed case in a dairy herd in Oregon. Oregon's dairy industry is both small and sequestered, and we rarely import cattle. So, what does avian influenza surveillance look like in Oregon? So, Oregon surveillance takes a multi-sectoral approach, which integrates wildlife surveillance, agricultural surveillance, and human health surveillance. And then additionally, we have integrated wastewater surveillance for the avian influenza H5 subtype. So wildlife surveillance in Oregon is again a collaborative effort. Oregon Department of Fish and Wildlife and the USDA National Wildlife Disease Program work with other federal and state partners to conduct surveillance testing on wild birds with a focus on waterfowl. This primarily includes morbidity and mortality surveillance, so the investigation of sick or dead wild birds, and watershed surveillance. So watershed surveillance being the testing of dabbling and diving ducks at specific watersheds. The watersheds are selected based on areas that have high mixing of wild bird populations, sometimes containing multiple flyways, and previous evidence of influenza infections in wild birds in the area. Samples can be collected from agency harvested birds, hunter harvested birds, and live wild birds, with target sample numbers for each season. The seasons being bird seasons, so summer being the end of breeding season, fall is around fall migration, winter is when birds are on their overwintering grounds, and spring is the spring migration and early breeding season. While wild bird surveillance is critical to understanding the distribution of avian influenza in U.S. wild waterfowl, it is dependent on convenient sampling. So sampling is often heaviest during the hunting season and does have some spatial and temporal gaps. Agricultural surveillance for avian influenza in Oregon is led by Oregon Department of Agriculture and includes responding to high-path avian influenza outbreaks in domestic poultry, managing regional quarantines and depopulations, additionally maintaining biosecurity practices at farms, the regulation of imported animals, and most recently, bulk tank milk surveillance. In December of 2024, the Oregon Department of Agriculture, with input from Oregon's dairy industry, including both operations and processors, launched its milk testing and monitoring program. By January of 2025, more than 75% of Oregon's milk was under surveillance. To date, Oregon has not detected H5 in dairy cattle or milk. This graph shows the number of samples tested between the start of the program and February 2025. Avian influenza surveillance of the people in Oregon is a collaborative effort between the Oregon Health Authority, Oregon Department of Agriculture, and the Oregon State Public Health Lab. All people in Oregon exposed to infected animals are monitored by public health through 10 days following their last exposure. And public health laboratory surveillance for avian influenza in humans includes the subtyping of all influenza specimens sent to the Oregon State Public Health Laboratory by outpatient emergency department and hospital settings. A subset of these specimens are also sent to the CDC each month for additional characterization. This is related to novel influenza virus detection, virus vaccine selection, and antiviral resistance. The sample size sent to CDC each month depends on the state and the time of year. So in the spring of 2024, with the emergence of high-path avian influenza in dairy cattle, there was a national interest in strengthening our existing surveillance systems. During the COVID-19 pandemic, wastewater surveillance had proved an important tool in the surveillance of emerging pathogens. So the CDC began collaborating with state and local health departments to expand their wastewater surveillance programs to include an avian influenza target. So primarily an assay that targets the avian influenza H5 subtype. However, for Oregon and most jurisdictions, this was the first time we were routinely surveilling a pathogen in wastewater that is endemic in an animal population. So to start out, as a quick overview of how wastewater surveillance works, to get everyone on the same page. First, an infected individual who has sewer utilities, so not a septic system, releases pieces of a pathogen or sometimes a chemical marker into the wastewater. This can happen with defecation, urination, and to a lesser extent, showering, hand washing, or clothes washing. Next, those pathogen fragments travel down the drain through the sewage system until they reach a wastewater treatment plant. So sewage pipe systems can be closed, so the pipes should only contain wastewater, or open, or also called combined, when wastewater is processed with stormwater. Although importantly, even closed wastewater systems contain stormwater through a process called inflow and infiltration, which is basically leaky pipes and water overflow. Then an operator at the wastewater treatment plant will take a sample. This can be a grab sample, but it's usually influent, which means wastewater as it's entering the treatment plant, but before it's been treated. And then some jurisdictions will sample from other locations, such as in clarifiers, which is where liquid and solids are separated. The sample is also often a composite sample, so it's collected by a machine in small amounts over a 24-hour period. Next, that sample is filtered to concentrate the signal and sent to a lab for usually PCR testing or sequencing to detect and quantify the amount of signal. And then finally, the results are reported to public health. Wastewater surveillance has several advantages over traditional surveillance systems. One of the most significant is a single sample represents an entire community. This also makes wastewater surveillance less biased in a number of ways. It captures mild and asymptomatic infections, and it's independent of healthcare seeking behavior or testing access. Often, wastewater surveillance provides more granular data. So, for example, surveillance on a city level or county level rather than on the state level. It can provide results in near real time, and it's relatively cost effective. Finally, it's adaptable to changing public health priorities. Once a wastewater surveillance system is in place, the locations and targets can be pretty quickly shifted. There are some disadvantages. Wastewater surveillance captures people on public sewer only, It requires wastewater treatment plant cooperation, which is almost always voluntary. And finally, there can be a lack of standardization across jurisdictions, which makes aggregating data at the national level more challenging. Oregon's wastewater surveillance program is a collaborative effort between Oregon Health Authority, Oregon State University, and 30 to 40 wastewater treatment plants from across the state. Our program covers approximately 80% of eligible Oregonians, so those on sewer, which is about 70% of Oregonians. In Oregon, 24-hour composite samples are collected from participating wastewater treatment facilities 1 to 2 times weekly. We only collect influent samples. And samples are tested with real-time digital PCR or dPCR for targets of interest, with additional sequencing for SARS-CoV-2 that have a positive detection with a concentration over a certain threshold. The samples are also archived for future testing. The interest in wastewater surveillance for avian influenza and the H5 subtype came at a time where Oregon was already routinely surveilling wastewater for SARS-CoV-2, influenza A and B, and RSV, and had conducted several special studies looking at wastewater concentrations of other pathogens on a smaller scale. We were also lucky that one of our program's principal investigators was working closely with Oregon Department of Agriculture at the time on the avian influenza response, and they theorized early on that we may see H5 detections in wastewater without cases in cows or people, but due to bird input into the wastewater. So in the spring and summer of 2024, as the CDC's National Wastewater Surveillance System, or NEWS, expanded their public-facing dashboard to include H5 detections for sites prospectively testing samples, Oregon decided to complete a retrospective study on the H5 subtype in wastewater. So the goals of this project were one, to establish baseline levels of the avian influenza H5 subtype in Oregon's historic wastewater samples, and two, to perform a descriptive analysis of H5 in Oregon wastewater to answer questions such as when does H5 enter the wastewater? Does the timing correspond to the emergence of the new genotype in Oregon birds or the spillover to US dairy cows? Do H5 detections in wastewater correspond with dairy processors or licensed operations within the sewer shed, or poultry outbreaks or wild bird density? Most importantly, we hope to use this information to inform our interpretation and communication of H5 and influenza A detections in wastewater prospectively. And we hypothesized that we would see wastewater detections of H5 due to bird input into the wastewater. Wild bird input gets into wastewater primarily through stormwater in open sewer sheds and through inflow and infiltration in all sewer sheds. But animal input can also enter wastewater through the dumping of contaminated animal products. And then finally, in jurisdictions that take wastewater samples from clarifiers, such as the picture on the left, birds are submitting their fecal samples directly for testing, like in that picture. So the sample population for this initial study included all archived influenza A positive samples from 20 current and historic participants in Oregon's wastewater surveillance program. Just as a note, wastewater surveillance for influenza occurred only during the influenza seasons for the 2021-22 and 22-23 seasons, but we transitioned to continuous surveillance in October 2023. Among the 551 influenza A positive samples tested retrospectively, 21, or about 4%, tested positive for the H5 subtype. You can also see the seasonal boundaries on our previous influenza A surveillance with those dotted lines on the graph. The first sample where H5 was detected in wastewater, that navy blue arrow above, was from a sample collected in March of 2022. This was about a month after H5N1 was detected in a wild bird in the United States. That's that first green arrow. and a month and a half before it was detected in wild birds in Oregon, that second green arrow. So we found that wastewater surveillance was an early indicator of the emergence of avian influenza in Oregon birds. Also, the first H5 subtype detection in Oregon wastewater occurred almost 2 years before the multi-state outbreak in dairy cattle. Those are the orange arrows representing the suspected timing of the spillover, the first spillover, and the first case in cattle. So this excluded cattle as a potential source for at least those first 2 years of H5 subtype detections. This is that same graph of influenza H5 detections in wastewater, Oregon wastewater on the top, with H5 detections in wild birds in the middle, and high-path avian influenza outbreaks in poultry at the bottom. You'll notice some seasonality in the wild bird detections, but just to note, this is partly due to convenient sampling around the hunting season. This is a breakdown of the H5 detections in wastewater by community. The 21 H5 detections were in 12 of 20 communities. The highest number of detections occurred in Ontario. The largest proportion of detections was in Newport. 8 of 12 communities with H5 subtype detections were within counties that previously had an outbreak of high-path avian influenza in poultry. Only 4 of 12 communities with H5 subtype detections had a licensed grade A dairy processing facility or dairy farm within the sewer shed. The H5 subtype was not detected in wastewater from the 2 sewer sheds with the highest number of licensed grade A dairy processing facilities, which is Portland with 7, or grade A dairy farms, which is Tillamook. There was no statistical association between H5 detections in wastewater and poultry outbreaks or the presence of licensed dairy processing facilities or farms within the sewer shed. Instead, we found that the 2 communities in Oregon with the most avian influenza H5 subtype detections contained important habitats for migratory wild birds, including seasonal wetlands in Ontario and an estuary of a major river in Newport. H5 subtype detections occurred in both combined and separate sewer sheds. Based on our conversations with Oregon Department of Agriculture, our findings were not surprising, partially because you can see on this map, these are Oregon sewer sheds that are along the I-5 corridor, outlined or filled in yellow, and dairy processors and dairy operations denoted with those black markers. Most licensed dairy operations in Oregon are located outside sewer shed boundaries, so they're not going to be captured by this surveillance system. And we learned with this study that wild birds are an important consideration when interpreting avian influenza H5 subtype detections in wastewater, and that we could expect continued intermittent detections of the H5 subtype in wastewater, even in the absence of outbreaks in dairy cattle or occurrence of human cases. We continued to monitor the H5 subtype prospectively in wastewater with this in mind. And in December 2024, we saw an increase of H5% positivity in wastewater. You can see the increase in percent positivity on that top graph and an increase in the concentration of H5 in wastewater samples on the right graph, starting in December 2024 and then peaking in February. The increased percent positivity coincided with the emergence of the D1.1 genotype of avian influenza in U.S. wild birds and the overwintering period for many Oregon waterfowl. This is the time when waterfowl are sitting on open water and likely contributing more significantly to stormwater. This was also a point when Oregon had started bulk tank milk surveillance, the graph on the bottom, and that helped provide further evidence that the H5 detections we were seeing in wastewater were not coming from Oregon cattle. While we still believe wastewater surveillance strengthens ongoing avian influenza surveillance efforts, we have learned that it needs to be interpreted with a consideration of all animal contributors and in conjunction with other surveillance metrics. This is the National Wastewater Surveillance System dashboard showing H5 detections for, I think this is the last 2 weeks of January, and we continue to monitor this weekly. So then as a final topic, I have a brief overview discussing the first case of the new genotype of avian influenza in pigs. So in October of 2024, clade 2.3.4b was detected in swine in Crook County, Oregon. The Oregon Department of Agriculture was investigating an outbreak of high-path avian influenza in backyard poultry. These included cooped chickens and free-range duck, geese, and peafowl. During this investigation, they identified three kune-kune pigs and two miniature pigs that were sharing living spaces with the poultry. A kune-kune pig was also observed scavenging a duck carcass. All of the pigs were asymptomatic. A pooled oropharyngeal swab from the kune-kune pigs was collected and tested positive for influenza A H5 on PCR. Just as a note, during this outbreak, most of the samples were tested with the three PCR assays listed here. For this presentation, I'm just referring to a positive result of the avian influenza virus subtype H5 PCR or the avian influenza virus subtype H5 clade 2.3.4 PCR as a detection of influenza A H5. On follow-up visits, individual nasal and oropharyngeal swabs were collected from the miniature pigs, and between October 25th and 28th, all pigs were euthanized for necropsy. There was detectable influenza A H5 in nasal and oropharyngeal swabs from a miniature pig and multiple necropsy samples from the kune-kune sow, lung samples from the kune-kune boar, a nasal swab from the kune-kune piglet, and multiple necropsy samples from one of the miniature pigs had detectable influenza A H5 on PCR. Incomplete sequencing due to low viral levels of one pig sample and full sequencing of samples from farm poultry and migratory birds in the area identified clade 2.3.4b, genotype D1.2. The public health response to the outbreak included poultry and swine depopulation and a 120-day farm quarantine. The remaining farm animals were monitored for 30 days. The farm was a non-commercial operation, and no animals entered the food supply chain. 6 people were exposed to infected animals and actively monitored by the local public health authority. Significant risks for cross-species viral transmission were identified during the investigation, including irrigation ponds on the property where wild and free-range poultry intermixed, and swine and poultry cohabitation. Despite clade 2.3.4b's high propensity to infect mammals, the risk of it entering the swine population was thought to be low. Experimental nasal or alimentary exposure of swine to the clade 2.3.4.4b led to marginal viral replication, with only 1 of 8 pigs exposed nasally seroconverting. While there was no indication of pig-to-pig transmission here in Oregon, this outbreak demonstrates that pigs are susceptible to spillover of clade 2.3.4.4b, and swine are an important intermediate host for influenza reassortment and antigenic shift, and pigs should be a biosecurity priority during high-path avian influenza response. With that, I'm sorry I went through this so fast, but I wanted to thank the people collaborating to perform avian influenza surveillance in Oregon. And if anyone wants to read the details about the 2 studies I talked about, you can find both of them by putting the terms Oregon wastewater and H5 into PubMed or Google and can read about the methods or results in more detail. And with that, I'm happy to answer any questions that people have for me or also Dr. Schultz.
Okay, we've got several questions here. I'm going to start off with the first question. It's another one that I had also. Um, it seems like the wastewater surveillance programs could have so many applications. Do you know what the decision-making process is for selecting pathogens to monitor? And my question, as an add-on, would be, do most states have programs for the same diseases, or does it vary by state?
Yeah, they're both good questions. You're probably not going to be surprised to hear that a lot of it is funding driven, and the CDC News does release priority pathogens that they would like wastewater surveillance monitoring for as part of their national program. Um, it is uh, looking at, if you go to the website, someone put it in the chat, the National Wastewater Surveillance System, you can see the national monitoring pathogens, which right now are SARS-CoV-2, influenza. They do influenza A only on the national site, RSV, Mpox, and if you go to the influenza A site and scroll down, that's where you find that H5 dashboard specifically. Oregon also has a state dashboard where we will publish results that aren't being published by the national dashboard. Oh, sorry, the news has recently onboarded measles as well. They display wild type measles detections, but a lot of jurisdictions also will test for vaccine-derived measles. So in your state, you also might find your state has an individual dashboard where they're displaying results that might be more granular than the National Wastewater Surveillance System.
Okay. Is it possible to track the source species, humans versus birds versus cattle, of H5 HPAIV detected in wastewater?
I mean, it's a really good question. So one of the things we've been working on for a long time is sequencing wastewater samples. This is fairly straightforward for which, if you sequence the sample, you're able to see at least looking at the databases, the genomic databases, what's the closest sequence and what animal host has it been identified in. But influenza A has been very challenging for us to sequence in wastewater samples because if you think about it, it's what's it called, where the virus is in pieces, a segmented virus. And so when you have a wastewater sample, you basically have a complex sample that's a bunch of different segmented viruses. So you might have like H5 subtype and N1 subtype, but they're not stuck together as like an H5N1 that you can sequence. Versus if you're sampling like a single person with a single infection, you're just sequencing that one subtype or clade or whatever. So it's been very challenging for us to develop, and for a lot of jurisdictions who have been working on this, to develop a bioinformatics pipeline for sequencing of avian influenza samples. So right now, we don't have a very good way to tell where our signals are coming from, other than from what I presented. Using our other surveillance systems combined with wastewater surveillance.
Okay. One of our attendees asks if wastewater samples, how long they're maintained. Are they maintained long enough to potentially do a retrospective study?
Yeah. So it's a really good question. So we freeze all of our samples, and this might be a better question too. I can always connect you with our laboratory PI. But we freeze all of our samples, and we find the biggest limiting factor is actually how many freeze thaw cycles we've put the archive specimen through. So we can go back to, samples that we collected at the beginning of our program, in 2021, and compare it. We'll often use the SARS-CoV-2 concentrations to compare the signal to make sure that we haven't seen sample degradation. But we find that once we've frozen and thawed the sample over twice, so 3 times, that signal degradation becomes significant. So mostly we just have to be very careful with our retrospective projects in prioritizing what we want to use those samples for and how many times we want to thaw them.
Okay. Um, Question here is if you have any recommendations for ongoing wastewater surveillance for AI in Oregon and in other states.
Yeah, we've had some interesting special studies here in Oregon that have been that I've found especially interesting. These have included surveillance for like Candida auris and multi-drug resistant organisms at the facility level. So looking at like hospital effluent, so wastewater as it comes out of the hospital, to see if we can improve our surveillance sensitivity for MDROs. We have done some other really interesting projects, kind of as a One Health veterinarian, like looking at cryptosporidium in wastewater. Oregon or Portland, Oregon drinks unfiltered reservoir water, which is very unique for a city. And so that was kind of a special project where we wanted to look at our crypto levels and see how they differed in Portland versus other places in the state. So I agree with the comment earlier that I think wastewater surveillance has like huge potential for a lot of pathogens. And yeah, it's mostly just interest and funding availability that's kind of our limiting factor.
Okay. Kind of switching species a little bit. And someone asked, whether there's any additional information on HPAI detection in cats and if there's any known human cases.
Dr. Schultz, that's a good one for you.
Yeah, I can take that one. So we have had quite a few cat cases. I was just pulling up the our case log. So it looks like we've had a total of 7 separate cases, with at least one of those having multiple cats. I believe if there was 7 or 8 cats in one of those cases, and I know the first case was multiple cats as well. So we have had one cat case. So there was a feline case back in 2022, December of 22, that we had that was associated with a backyard poultry premise that was infected. Those cats resided and were co-located in a chicken coop with the chickens that were infected. And then we've had a number of domestic cat cases since then, kind of tied to a couple of different raw pet food outbreaks. And then one additional cat that we believe was exposed was a feral cat that was living right on the edge of a wildlife refuge. We believe was likely environmental exposure on that one. Human case wise, I'm probably not the authority on this one, but we have had one human case that we confirmed to be related to an exposure that occurred in Oregon as part of a commercial poultry response. There were a few other positives that were human positives that were test positive, but they tested positive on entry into the farm. They had moved from farm to farm and had, like we believe, had been exposed at a previous farm, as they hadn't been working on the farm here in Oregon yet when they tested positive.
Okay. And in the chat, one of the attendees says that there's been a couple of domestic and wild felid detections in Washington as well. The individuals with the Washington State Department of Agriculture. There's also been a few occupational health exposures, but the risk to public remains low.
And then I can respond to the comment above that too. Um, Somebody commented that there are some biological source markers that you can look for that are human versus mammal versus avian specific, to help differentiate source. And this was something that we also considered as well, like looking at, they have like a gull, duck, goose marker that you can test for in wastewater. So our thought was, could we track the concentration of like a gull, I don't know why that's a tongue twister for me, gull, duck, goose marker. And as it goes up, if we see more H5 detections, we can kind of do a correlation analysis to say, okay, this is following the trend, that sort of thing. We have one of our graduate students at Oregon State University who's looking at exactly that study design.
Okay, kind of along the lines of the other species, is there any potential to get poultry or swine processing plant wastewater? You concentrated mostly on cattle facilities, but any move towards other species, processing plants or facilities?
Yeah, I can take that. I start was typing a little bit into the chat there too on that one, but not at this point. Part of that is because Oregon is somewhat unique, that while we do have a fair amount of commercial poultry production, we do not have any processing plants in the state. Those birds all get processed up in Washington. But as a part of that production cycle, every one of those flocks is tested prior to processing. And so we have a really good consistent surveillance stream coming from those samples all get tested and reported to us. In terms of swine, again, we don't have much. We have one processing plant here that's classified as a very, very small plant. And the about 90+% of what they are processing is coming from out of state, and most of it actually from hunter-rat colonies up in Canada. So not really something we've been looking at. Again, I think that if the interest is to do surveillance on those species, it's probably simpler and more accurate to just do the surveillance to do the direct surveillance and collect the samples that would be the ideal samples rather than trying to look at targeted effluent testing.
All right. Tindy asks, regarding the specific laboratories that Oregon uses or has used for feline cases for H5 testing, a state lab or
Yeah, all of those have been going through our NALM lab here at Oregon State University. That's part of the Oregon Veterinary Diagnostic Laboratory there.
okay. Someone asked, and I don't know if you would have this information, but any emerging trends in data as to the geographic distribution and frequency of measles based on wastewater surveillance?
I think what I can say is we have recently published an article on measles detection in wastewater, and what you can again just look it up by clicking measles wastewater Oregon. And what we did is we actually did another retrospective study, which is something we love to do before we start prospective surveillance so that we can inform ourselves on what a kind of baseline will look like. But what we asked was kind of what would we expect measles in wastewater to look like around an outbreak for this particular one. And in 2024, Oregon actually had a pretty, for us at least, a significant measles outbreak for us at the time. It was like 31 cases over 2 counties, or with 30 of the cases in 2 counties. And so we looked back retrospectively and tested wastewater and found that we had detections of measles for like 10 weeks before the first case was identified using, case finding, case surveillance. We also saw that the concentration went from a really low level of measles in the wastewater and then spiked to really high concentrations during the start of the outbreak. And then the signal in wastewater disappeared. And after the signal disappeared in wastewater, we actually had another 8 cases identified after that, which we think might be related to the fact that a lot of the outbreak was off sewer shed boundaries. So we learned a lot from that retrospective study, and I think we're going to continue to learn what our prospective detections of measles mean in wastewater, but it's ongoing learning process.
Okay. Few more questions. They keep kind of coming in. Dr. Scholes was typing an answer to this one. Seems rare, although possible, to have swine spillover when they are co-housed with poultry. Is there a way to rule out oral and nasal contamination on an infected farm?
Yeah, I was typing and it was getting really long. Thank you for jumping in on that one and letting me do it verbally. So my bias is going to show here a little bit, I think. But to me, as the state veterinarian, as the one in Oregon who's responsible for making these decisions, I'm not sure I care. Right. There's a scientific distinction to be made between oral contamination and clinical infection. But to me, we know that pigs are a critically important host species for influenza, right? My virology is not the best, but I remember that from day one of virology, right? You don't let pigs combine with poultry influenza viruses. So to me, if we have a pig that is, even if it's oral contamination, we know that oral exposure is a route that can and now has led to clinical infection. I think we likely euthanized those pigs before there was really a chance to see what that virus truly was going to do, right? We were less than 96 hours after the initial exposure when we euthanized those pigs. But even then, the sow that consumed the duck that had the likely infected duck, it was systemically infected. We saw virus in organs and in systems that are not connected to the alimentary canal. So we know that there was systemic infection there. So for me, I'm not sure that I'm willing to give it the chance. We know that it's possible. I think it's our obligation to treat a pig that tests positive as a pig that is positive. If it's a situation where we can quarantine them safely, maybe we do that. But I I think we would, for speaking from on behalf of Oregon, I think given that situation and given that scenario or a similar scenario, we would do the same thing again, and we would indemnify and euthanize those pigs.
Okay. Kind of back on the measles, you spoke of a study in the data. Has that been published yet, Dr. Fallender?
Yeah, I can put the link in the chat. Give me a second.
Great,
Just don't want it to pop up on my shared screen here.
great. Someone noted that all of the pathogens currently under the surveillance program are viruses. Do you see any utility for complications to using wastewater to look for bacterial pathogens?
Yeah, we did a little bit of it with the multi-drug resistant organisms, and found it challenging. Yes, it just the general. I wasn't as involved in that project, so I think if you have specific questions at it, I'm happy to uh, kind of connect you with our laboratory person. But yes, we found that the bacterial monitoring was more complicated for several reasons. One of them being that the bacteria grows in the sewer pipes. And so we were dealing with detections that we thought were likely environmental pathogens that are just sitting there. So yeah, I don't think we're eager to look for more bacterial targets just right now. That being said, it's certainly we haven't, we didn't call it a failure, but it was more complex than viruses.
Okay. This one may be Dr. Scholz's. I don't know regarding vaccination of domestic poultry being considered as a management strategy versus depopulation.
Yeah, I mean, I think the short answer to that is it's complicated. There's probably others on this call that could answer this with a lot more authority than I can. Ultimately, that's going to be a federal decision. The vaccine is a controlled vaccine. And, anytime we start vaccinating for foreign animal diseases, it complicates our ability to ever declare freedom. It complicates trade. There's just a whole bunch of issues that come in there. Not the least of those issues is that the vaccines we have are not perfect vaccines. Some, like a lot of viral vaccines, they do not necessarily protect against 100% of infections. They do a really good job of reducing shedding. The problem with that is right now, our active surveillance for this virus is really based on mortality, especially in commercial farms. And so, in order to do that, it's not to say that we can't or shouldn't, but if we begin vaccinating, we will have to necessarily change our surveillance processes to be able to do that. Again, I think those are decisions that are probably being made way above my head, and I am perfectly happy for them to stay there, just because of all of the trade implications and the geopolitical things that go into that. So,
Okay, great. And the last one here is kind of a question. I don't know if it really has anything to do with wastewater surveillance, but someone in Delaware, there's a large pond behind their home. Residents have been asked to collect eggs of birds by local authorities. Are you familiar with any detection or any kind of surveillance based upon egg sampling for avian influenza?
no, and this is an answer I'm going to get myself in trouble on pretty quickly, I think. But the yeah, I don't know of any among that, being done that way. We certainly do see there is a lot of surveillance done in wild birds, typically as part of nesting grounds or hunter harvest. Those are typically swabs, oropharyngeal or cloacal swabs of the birds themselves. So I'm not aware of any. I'm also not really sure about the saltwater soak, what that has to do with it, or would have to do with influenza virus either. But yeah.
Okay, well, somebody in the chat commented that Canadian researchers are testing wild bird eggs. So apparently it is happening. Okay, I do not see any additional questions. I appreciate all the questions that have come in from our attendees. I want to thank our two guest speakers today, Dr. Founder and Dr. Schultz, for a very engaging and informative presentation. We can't do this without volunteers such as them. So definitely, if you are interested in providing such a presentation, or if you know someone, encourage them to contact us, and we can get you in the lineup. Upcoming presentations in March, we'll announce it pretty quickly and send out an invitation. We have a presentation from Dr. Lonnie King on One Health. And then in April, we have another infectious disease topic from a speaker we've had before. But we do this every month, but we can't do it without volunteers such as Dr. Founder and Dr. Schultz. So thank you to our speakers. I want to thank all of our attendees, and I appreciate everybody's time. Everybody have a very safe and happy evening. So with that, I will close out the webinar. Thank you very much.